Hi, my name is Kaspar Vogt, and I'm a PI at the WPI IIIS, the Sleep Research Institute at the University of Tsukuba. And for today's video, I would like to explain you a little bit what we are doing here in the lab and why we are doing it. We all need sleep. You notice this several times a week, probably, that you get tired and that you need sleep. And as you can see here from this illustrious panel of people, we don't always get the amount of sleep that we need, and you can see them here nodding off during the day. Now, although we don't know exactly why we need to sleep, we certainly know that we do need. And if you don't sleep enough, you get into all sorts of problems. And sleep is not just switching your brain off, as I will try to explain to you in the following slides. Very interestingly, things are happening in our brains during sleep that improve its function, that enhance memory, for example. And this has been known for a long time. Already the old Romans knew that if you were rehearsing a speech, that sleeping over it for a night would enhance your memory and improve your thinking about things. So sleep is not just something passive, and we know that also from recording brain activity, especially cortex activity, the outermost layer of the brain. And when people electrically probe this, what they find is that in waking, you have highly irregular, desynchronized activity in cortex. And then when you fall asleep, you can see this in the bottom two lines on this slide. The top of these two lines is the so-called EEG. This is the electroencephalogram. This shows us the activity in cortex as a sort of summary. And the line below is the so-called EMG. This is muscle activity. And you see here a recording from an animal that is just about to fall asleep. So you see that it relaxes its muscles, and in the EEG you can see this transition from this highly irregular activity in waking to a more regular, slow activity in sleep. And this is well known, this has been studied, and it was found that these slow waves are caused by large numbers of neurons going from a hyperpolarized silent state to a depolarized and active state together. And that gives rise to these slow oscillations in the EEG. And this activity matters because to this day, we still don't fully understand how, for example, memory is improved in the brain during sleep. And there are two hypotheses. They are not necessarily competing, but they're still quite different. The SHI hypothesis, the synaptic homeostasis hypothesis, postulates that during waking, all activity leads to a strengthening of synapses, and that strengthening needs to be reduced somehow during sleep. So this is a very global process, whereas some evidence has been produced that there is a so-called replay process going on in the brain during sleep, meaning that when you have a certain memory being formed, there's a certain number of neurons that are active together, and these are reactivated, and that strengthens the memory. And this is a very specific process. This is something that affects primarily the neurons that were active during waking. So it is useful and necessary for us to understand precisely what's going on in the cortex. And this is what we do. So these are so-called tetrodes. These are bundles of very, very thin wires, of 4 wires that are lowered into the cortex into the brain. You can see on the right-hand side a schematic from a publication by Bujaki, where you can see where the tip of these tetrodes is embedded in the neurons that it records from. And we can extract two different signals from these tetrodes. The one is called LFP, local field potential, and this is the top trace here in the bottom half of the slide. And again, you see that this local field potential looks very different between the waking state of the animal and the sleeping state of the animal. Again, we have a kind of high frequency irregular activity during waking, and we have slow waves during non-REM sleep. Interestingly, we can also extract so-called unit activity. We can extract spikes of individual neurons that are very, very close to these four wires, and each neuron firing an action potential is then marked as a small dot on the graph. And what you can see is, again, that in waking, the activity of these units is highly irregular and unpredictable and not synchronized. And as soon as the animal transitions into non-REM sleep, these neurons become synchronized. They are active together, and then they're silent again, and then they are active again, and they're silent again. And all this is synchronized so that we can actually see this in the local field potential. But now we want to understand what's going on a little bit further. So is this just kind of idling activity? Is just everything slowing down, and that's the most efficient way of doing that? For this, we simply looked at each individual neuron and looked at its activity before the animal was falling asleep and then when it was in the sleep phase. And very interestingly, we see that falling asleep has very different effects on different types of neurons. If the neuron is very active during sleep, then its activity is considerably reduced. You can see this in the gray bars in these graphs. So the top sextile is the most active neurons. Their frequency all goes down. Pyramidal neurons are neurons that are excitatory. Interneurons are neurons that are inhibitory. And for both of these types, falling asleep means going down if your wake activity was very high. However, if your wake activity was very low, then your activity actually goes up when you fall asleep. And that argues against a simple energy preservation mechanism for sleep in the cortex, because there would be no reason and no use to increase the activity of any neurons. You would reduce activity across the board to make sure that you saved as much energy as you can. What we then looked at is simply when we had a number of neurons that we could identify, we could look at their data stream. We could look at which neuron was active after which other neuron and so on. So this gives you a kind of pattern. You can try to predict how these work together. And there is a simple measure for the organization in such a pattern, and that's the entropy. You have the formula here. It doesn't matter too much precisely, but what we can do is now look at different patterns of activity and how well organized they are. And we consistently find that the entropy goes up in the transition from the waking state to the non-REM sleeping state, the non-rapid eye movement sleeping state, meaning that the predictability of these patterns, the organization of these patterns goes down when the animal falls asleep. So if there is replay, for example, then at least it's not the dominant thing going on during non-REM sleep, because the organization is actually lower in non-REM sleep compared to waking. Whereas if the brain was simply repeating a few patterns over and over and over during sleep, you would expect a much higher predictability of these patterns. And so for the past few slides, we have simply looked at the spontaneous activity of the brain, and that gives you some insight. So we see that certain neurons go up in their activity, certain neurons go down in their activity. The entire organization is less tight in non-REM sleep compared to waking, but we need other measures to probe the cortex during sleep. And what we have done here is, in the top left corner, you see a schematic of the brain, a so-called coronal slice, and you see we injected a virus into a part of the brain that projects to the cortex, that sends excitation to the cortex. And this virus makes neurons express channel rhodopsin, a light-sensitive excitatory ion channel. So we can excite this input to cortex with blue light. This is called optogenetics. And then we can study the response of the cortex to this excitation in all different states, because we can tether this animal chronically to an apparatus that will let it allow to walk around freely, but that will let us allow to send laser pulses to this brain and record its electrical activity. And on the top right side, you see now the recording from one of these tetrodes, and this is the local field potential. So this is kind of the sum of activity that you get. And the green arrows are every time we send a brief laser pulse into this brain. And you can see that down below in blue, slow wave sleep or non-REM sleep, the responses are much, much bigger. Very, very surprisingly, in wake, cortex reacts much, much less to these pulses compared to non-REM or slow wave sleep. And the bottom of the slide shows you a 24-hour recording of an animal. Ipsilateral is the tetrode that is on the same side as the light stimulation. Contralateral is on the opposite side. So this excitation actually spreads quite a lot over the cortex, especially during non-REM sleep, during slow wave sleep. You can see here that there is a very clear distinction between the response in slow wave sleep, in non-REM sleep, and waking. And very, very surprisingly, it's the opposite of what many people would have suspected. So the reactivity of the brain, the response to a defined stimulus, is much larger in non-REM sleep compared to waking. So it's as if something in waking would prevent this kind of runaway excitation to occur. And we are still working on the exact circuitry that underlies this, but we do think that this is very, very important in our understanding of how cortex sleeps. This is a summary of a number of animals that were studied, and again, you see here that the response to the stimulus is much, much smaller in the waking state and grows very, very large in slow wave sleep, and is somewhat smaller in rapid eye movement or REM sleep. So compared to waking, something seems much more reactive. And our understanding of how the brain can change between these two modes will be very, very important in understanding how the brain fulfills its function, fulfills its memory enhancing function, but also its other functions that make you from being very, very tired to relaxed and awake again the next morning when you have a good night's sleep. So to summarize these things, in non-REM, non-rapid eye movement sleep, neurons in the cortex are highly synchronized and they produce these slow waves. And we still don't know exactly how they are synchronized, and we certainly don't know why. And what we know, however, that certain neurons increase their activity and certain neurons, especially highly active neurons, decrease their activity in the transition to sleep, meaning that this is not just a global shutdown of cortex. This is a very, very specific reorganization of cortical activity, and we want to understand how precisely this reorganization affects brain function. We know that the patterns that we record during sleep are a lot less organized than what we see in waking. And it's easy to speculate that, in waking you have to process information. What you do with your brain when you're awake is dictated by your environment. And then when you fall asleep, the entire input from the environment goes way, way down. And now your brain is left to its own devices, but it's still active. It's still producing activity. And what this activity, which is somewhat less organized than in wake, is doing, that's what we're trying to understand. And again, surprisingly, we get a higher evoked response in non-REM compared to waking. And we are now working on trying to unravel why this response grows larger. And this will hopefully let us understand how this very, very dynamic shift in brain activity happens and why it happens in the transition from waking to non-REM sleep. Thank you very much.